Hi everybody and welcome to our module on glycolysis. Glycolysis is a very ancient way of generating energy for cellular metabolism. It was probably one of the first methods of generating energy used by life when it formed on Earth. And in the human body, it's used by all cells. It's a sequence of reactions that occurs in the cytoplasm of cells, and it converts glucose, which as you may know has 6 carbons, to pyruvate, which has 3 carbons. And in the process of doing this, it generates ATP and NADH. Now, most of you probably know that ATP can be used by cells for energy, but you may be less familiar with NADH, so let's talk about that now. NADH stands for nicotinamide adenine dinucleotide, and just like the name implies, it is 2 nucleotides joined together. If we look at the chemical structure of NADH on the screen here, you can see at the bottom that we have a 5-carbon sugar here, a phosphate group here, and we have a base. So this makes this a nucleotide, just like the nucleotides found in DNA and RNA. And there are 2 nucleotides joined together by their phosphate groups in NADH. The base here on the bottom molecule is adenine, so this makes this a molecule of adenosine at the bottom. The base at the top here is nicotinamide. Nicotinamide is not used in DNA or RNA, but it is found in molecules of NADH. You can see that in this molecule I've drawn here, the nitrogen atom in nicotinamide is not protonated. It has a positive charge. So that makes this a molecule of NAD+. If there were a proton attached here, this double bond would go away, and then we would have a molecule of NADH. The function of NADH in cellular metabolism is to carry electrons. So NAD+ can accept electrons. NAD with a hydrogen attached can donate electrons. And you can just remember that anytime the hydrogen is attached, it is carrying electrons that it can donate for other reactions. And one of the most important places that NADH donates its electrons is to the electron transport chain in the mitochondria. This leads to the generation of ATP. Shown on the screen here are the reactions for glycolysis, and you may have learned about these in a college course in biochemistry, but for medical school and USMLE Step 1, it's very low yield to memorize the names of all of these molecules and all the enzymes involved. Only certain ones are important, and we'll go through these in this module now. Some key things to recognize are that glucose, as you may know, is a 6-carbon molecule. However, at this stage of glycolysis, the glucose molecule is split into 2 3-carbon molecules. This is the structure of glyceraldehyde 3-phosphate on the screen here, and you can see that it only has 3 carbons in comparison to glucose, which has 6. The remainder of the reactions involve 3-carbon structures, and the sequence finishes with pyruvate, which is a 3-carbon structure, as I've shown on the screen right here. Another key point to recognize is that most of these reactions are reversible, and thus I've shown them as 2 green arrows going in both directions. This means that when the level of one substrate gets high, for example, if 3-phosphoglycerate got high in this reaction right here, then via equilibrium we would generate more 2-phosphoglycerate. On the other hand, if 2-phosphoglycerate got high, then via equilibrium we would generate more 3-phosphoglycerate. So these reversible reactions can go in both directions. Therefore, they are used in glycolysis to go this way and generate pyruvate, but they are also used in gluconeogenesis to go the other way and generate glucose. And I talk about gluconeogenesis in a separate module. 3 of these reactions, however, are not reversible, and they are indicated by the red arrows on the screen here. So the conversion of glucose to glucose 6-phosphate is not reversible, fructose 6-phosphate to 1,6-bisphosphate is not reversible, and phosphoenolpyruvate to pyruvate is not reversible. These are the most important reactions to know for glycolysis, and we'll talk about these in detail in this module. Glycolysis is traditionally divided into 3 stages, and that's how we'll talk about it in this module today. The first stage is called the priming stage. This is where the cell invests energy and spends ATP in order to convert glucose to a more favorable structure to go through the remaining reactions. The second stage is called the splitting stage, where it divides from a 6-carbon structure to a 3-carbon, and then finally we have the energy stage where ATP is generated. The first reaction in the priming phase is the conversion of glucose to glucose 6-phosphate, and there are different enzymes in different cells in the body that catalyze this step. Some cells of the body use hexokinase and other cells use glucokinase. And just a side note here, in glycolysis and gluconeogenesis, anytime you see the name of the molecule have the word phosphate added to it, as in this step, then the enzyme that catalyzes that step is going to be a kinase. Kinase enzymes add phosphate groups, and most of the time that phosphate group will come from ATP. One of the things you need to be aware of to understand this step of glycolysis is that glucose often moves in and out of cells, but once it is phosphorylated to glucose 6-phosphate, it is trapped inside of cells. So hexokinase will catalyze this step for most tissues in the body. And hexokinase, in contrast to glucokinase, is strongly inhibited by glucose 6-phosphate. This blocks cells from hoarding glucose. Let me explain what I mean by that. Cells will take up glucose and phosphorylate it, but once they develop a sufficiently high level of glucose 6-phosphate, 6-phosphate that will inhibit this step, and the cell will stop taking up glucose. In other words, cells will only take up glucose until they have satisfied their metabolic requirements. Also, in contrast to glucokinase, insulin does not have an effect on hexokinase. It is always operating based on the level of glucose 6-phosphate inside the cell. And then the enzyme kinetics of hexokinase and glucokinase are different, and you need to understand those differences. So we'll talk about hexokinase first. It has a low Km, and this means it usually operates at the maximum rate. I'll talk about that in a minute. And it has a low Vm, which means that the maximum rate the enzyme can function is not that high. So to understand the Vm and the Km, we need to review a little bit of Michaelis-Menten kinetics. I discuss these in more detail in some of the basic pharmacology modules. So the Michaelis-Menten equation, written at the top of the screen here, says that the velocity or speed of a reaction catalyzed by an enzyme is proportional to the Vmax for that enzyme times the substrate concentration divided by Km, the Michaelis constant, plus the substrate concentration. So if Km is very low, as it is for hexokinase, then the Km will quickly become irrelevant as the substrate concentration rises. When the Km becomes irrelevant, this equation reduces to V equals Vmax. What this means is that the concentration of glucose only needs to rise just a little bit, and then this enzyme will immediately begin operating at Vmax. And that's how hexokinase works. It just takes a little bit of glucose to get it to operate at its maximum velocity, the maximum speed at which it can convert glucose into glucose 6-phosphate. Now, another feature of hexokinase, which is not readily obvious on this slide, is that it has a relatively low Vmax compared to glucokinase. In a minute, I'll show you a comparison of the two reactions, but this maximum velocity, maximum speed of the conversion of glucose to glucose 6-phosphate is lower than it is for glucokinase. Now let's talk about glucokinase. Glucokinase is found in the liver and also in the pancreas, and it has a couple of important differences from hexokinase. First of all, it is not inhibited by glucose 6-phosphate, and it is induced by insulin. In fact, insulin can promote transcription of this enzyme at a genetic level. Now, even though it isn't inhibited by glucose 6-phosphate like hexokinase, it can be inhibited by fructose 6-phosphate, which is the next molecule down the line in glycolysis. This inhibition, however, can be overcome by high levels of glucose, and let's talk about the implications of that. This means that any time the glucose level is high, glucokinase will be taking up glucose into the liver and converting it into glucose 6-phosphate. It doesn't matter what the level of fructose 6-phosphate is. If glucose levels are high, the liver will take up glucose. And that's because you want the liver to take up glucose anytime blood sugar levels are high so that it can store it as glycogen for later use. Another way to look at this is that the only time this enzyme is inactive is when you have two things present, both a low glucose level and a high level of fructose 6-phosphate. So let's think about this. When you're fasting, your blood sugar level becomes low. And also in times of fasting, the liver begins to perform gluconeogenesis. This means that pyruvate molecules are being converted into fructose 6-phosphate as part of gluconeogenesis. So when both of these are present, you want to shut down this enzyme so that glycolysis will not proceed and the liver will favor gluconeogenesis. Make sure you think about this. This inhibition by fructose 6-phosphate, which is overcome by high glucose, can be confusing. Another important difference between hexokinase and glucokinase has to do with the enzyme's kinetics. Glucokinase has a high Km, and this means that its rate varies with the concentration of glucose. So let's review the enzyme kinetics of glucokinase. It has a relatively high Km. This means it takes a high level of substrate concentration, a high level of glucose, in order for the Km to become irrelevant and for this equation to reduce to V equals Vmax. This means it takes a lot of glucose before glucokinase will reach its Vmax. And you can see I've shown that on the curve here. Hexokinase, which is the dotted line, reaches its Vmax right away at a relatively low concentration of glucose. You have to get to a much higher concentration of glucose in order for glucokinase to reach its Vmax, and that's what happens when the Km value is high for an enzyme. In addition, as you can see, the Vmax for glucokinase is much greater than the Vmax here for hexokinase. This means it can convert much more glucose to glucose 6-phosphate if the substrate concentration is sufficiently high. And then a final feature to know about of glucokinase is that it has a sigmoidal curve. This means the enzyme demonstrates the phenomena of cooperativity. Once it binds its first molecule of glucose, it then becomes more active and can more rapidly begin to ascend towards its Vmax. So I've summarized these points for you here. Glucokinase has a high Km. That means it takes longer for it to reach its Vmax, and it has a high Vmax, which means it can operate at a higher velocity compared to hexokinase. And as I've indicated up here, you might reach your Vmax after a meal when the glucose concentration is very, very high. Glucokinase will be very active, pulling lots of glucose into the liver, and that's because the liver wants to store it as glycogen for later use. There is an unusual mechanism by which fructose 6-phosphate inactivates glucokinase, and that's by way of a protein called glucokinase regulatory protein, or GKRP. This protein translocates glucokinase to the nucleus, and once the enzyme has been moved out of the cytosol and into the nucleus, it is inactivated. In the presence of fructose 6-phosphate, GKRP will bind to glucokinase and pull it to the nucleus, and I've shown that on this picture down here. GKRP is binding glucokinase in the presence of fructose 6-phosphate and pulling it to the nucleus where it is inactive. In the presence of glucose, however, glucose will compete with GKRP for GK binding, and when glucose is around, it will bump the GKRP off of glucokinase and release glucokinase so that it can go out into the cytosol and do its job. And so just like I showed you 2 slides ago, what this means is that the only way glucokinase is going to be inactive is when you have both the presence of fructose 6-phosphate and the absence of glucose. In other words, a low glucose level. These 2 features together will lead to the inactivation of glucokinase. Otherwise, if there's glucose around, no matter what fructose 6-phosphate tries to do, glucokinase will make its way to the cytosol and be active. So another way that can help you keep the functions of hexokinase and glucokinase straight is to think about how they operate in the settings of low blood sugar and high blood sugar. So when you have low blood sugar, for example, in the fasting state, hexokinase found in most tissues of the body is still working. There's a relatively low level of glucose 6-phosphate, and therefore hexokinase is phosphorylating glucose whenever it can get its hands on it. On the other hand, glucokinase will be relatively inactive. Its rate is proportional to the glucose concentration. Also, it is activated by insulin, and insulin levels will be low in the fasting state. This means that in the fasting state, when there's relatively little glucose around, glucose will go to tissues when they can find it, and the liver will not take any of it up. And that should make sense to you because, as you may know, the liver performs gluconeogenesis in the setting of fasting, and therefore it doesn't want to be phosphorylating glucose and taking it into glycolysis. On the other hand, after a meal, when the blood sugar level is high, hexokinase in tissues will be relatively inactive. That's because the level of glucose 6-phosphate will become elevated, and that inhibits this enzyme so that the molecules of glucose won't be hoarded by tissues in the body like your muscles. Glucokinase, on the other hand, in the liver will be working. There's a high glucose, and remember, high glucose tells glucokinase to operate at its maximum rate. There's also high insulin, and insulin can stimulate this enzyme. This means that in the setting of high blood sugar after a meal, the liver will store glucose as glycogen, and the tissues will take up only as much as they need in order to satisfy their metabolic requirements. Rarely, people can be born with a relative deficiency of the glucokinase enzyme, and this results in hyperglycemia. The liver takes up less glucose after a meal, and in addition, remember that glucokinase is also found in the pancreas, so the pancreas will be less sensitive to glucose, and therefore it won't release insulin as readily as it normally would. Generally, the symptoms are relatively mild. People with a glucokinase deficiency have a mild hyperglycemia. When the glucose level gets high enough, the pancreas does react and release insulin. One thing that you should know is that this deficiency often is exacerbated by pregnancy, and many women who are diagnosed with gestational diabetes may in fact have a relative glucokinase deficiency. Now let's talk about the next important reaction in glycolysis, and that is the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. This reaction is catalyzed by the enzyme phosphofructokinase 1, and it is the rate-limiting step for glycolysis. And note that we go from a molecule with 1 phosphate, that's fructose 6-phosphate, to a molecule with 2 phosphates, fructose 1,6-bisphosphate. And just like I said before, when you're adding a phosphate, it usually comes from ATP, and it's usually catalyzed by an enzyme with the word kinase in the name. If we go back to our overview slide, we previously talked about the conversion of glucose to glucose 6-phosphate. This is a reversible step here, so now we're going to talk about this step right here. It is the second irreversible reaction of glycolysis. This step consumes the second ATP in the priming step. As I said before, it's irreversible, and once glucose progresses through this step, it is committed to glycolysis. Other offshoots of glycolysis, like the HMP shunt or glycogen synthesis, are no longer possible. It is going to go all the way through to pyruvate. So this step controls the rate of glycolysis. It's the rate limiting step, and you know that glycolysis generates energy. Therefore, it should make intuitive sense to you that this step is inhibited by things that represent a high energy level in the cell. Citrate comes from the tricarboxylic acid cycle or the citric acid cycle. So when levels of citrate are high, the cell has plenty of energy, and therefore it's going to want to inhibit this step and slow down glycolysis. When ATP is high, obviously the cell has lots of energy, and therefore it wants to inhibit this step and perform less glycolysis. On the other hand, key inducers of this step are things that represent the need for more energy in the cell, the need for more glycolysis. So when AMP levels are high, recall that AMP is a breakdown product of ATP. So when AMP levels are high, lots of ATP is being broken down, and therefore the cell needs more energy, so it wants to activate this step to perform more glycolysis. And then a second important activator of this step is a very important molecule called fructose 2,6-bisphosphate. And this molecule is the way in which insulin and also glucagon control the rate of glycolysis. So let's talk about how the level of fructose 2,6-bisphosphate inside of cells regulates glycolysis. The conversion of fructose 6-phosphate to 1,6-bisphosphate is catalyzed by the enzyme phosphofructokinase 1. This is the step of glycolysis that we're talking about now. The reverse reaction in gluconeogenesis is catalyzed by an enzyme called fructose 1,6-bisphosphatase 1. Just like PFK1 is the rate-limiting enzyme for glycolysis, fructose bisphosphatase 1 is the rate-limiting enzyme for gluconeogenesis. This means that the relative activity of these two enzymes determines whether molecules will flow in this direction for glycolysis or in the reverse direction for gluconeogenesis. What fructose 2,6-bisphosphate does is it activates phosphofructokinase 1 and it inhibits fructose 1,6-bisphosphatase 1. This means that it drives the flow of molecules in this direction, in the direction of glycolysis. The easiest way to remember the function of fructose 2,6-bisphosphate is to think of it as an on-off switch for glycolysis. When its level is high, it favors glycolysis and turns glycolysis on. When its level is low, it favors gluconeogenesis and turns off glycolysis. What's confusing about fructose 2,6-bisphosphate is that it is synthesized from fructose 6-phosphate, one of the molecules that it is supposed to regulate. The way this is done is by a pair of enzymes called phosphofructokinase 2 and fructose 1,6-bisphosphatase 2. They have the same name as these two enzymes here, they just have a 2 instead of a 1. And these two enzymes control the conversion of fructose 6-phosphate to 2,6-bisphosphate and the reverse reaction as well. So as we will see on the next slide, insulin or glucagon can result in the phosphorylation or dephosphorylation of these two enzymes right here. And depending on whether they are phosphorylated or dephosphorylated, they will tend to go either in this direction or in the opposite direction, and that is how insulin and glucagon can raise or lower the level of fructose 2,6-bisphosphate, which in turn affects the activity of these two enzymes here. This is a very confusing system, but if you just remember what I've written in this box here and think of 2,6-bisphosphate as an on-off switch for glycolysis, you can usually figure out what's going on at the enzyme level. There's a complex second messenger system whereby insulin and glucagon regulate the levels of fructose 2,6-bisphosphate inside of cells. As I've shown here, molecules of insulin or glucagon can interact with surface receptors and manipulate the function of adenyl cyclase. Insulin inhibits adenyl cyclase and glucagon activates it. Adenyl cyclase increases levels of cyclic AMP inside of cells, and cyclic AMP activates protein kinase A. Protein kinase A will then phosphorylate phosphofructokinase 2 and fructose bisphosphatase 2, and this is how the level of fructose 2,6-phosphate will be manipulated. So what I just showed you on the last slide is that glucagon leads to phosphorylation of these two enzymes right here. Insulin leads to dephosphorylation of these two enzymes here. And by doing that, that's the way insulin and glucagon manipulate the levels of 2,6-bisphosphate inside of cells. Because this second messenger system can be confusing, I'll give you two more slides to hopefully make it very clear and stick in your mind. Insulin leads to dephosphorylation of these two enzymes. This favors the conversion of fructose 6-phosphate to 2,6-bisphosphate and increases the levels of fructose 2,6-phosphate. This goes and turns on glycolysis and therefore activates phosphofructokinase 1 so that glycolysis will proceed. You can see this man is eating, his insulin levels are high, and therefore his fructose 2,6-bisphosphate levels are also high. In the fasting state, represented by this empty refrigerator over here, glucagon leads to phosphorylation of these two enzymes right here. And when they are phosphorylated, this favors the conversion of fructose 2,6-bisphosphate back into fructose 6-phosphate. This will lower the levels of fructose 2,6-bisphosphate, which in turn will inhibit phosphofructokinase and glycolysis and favor the reverse process of gluconeogenesis. So that concludes our discussion of the priming stage of glycolysis. Now let's talk about the splitting stage. This is where a molecule of fructose 1,6-phosphate is converted into two molecules of glyceraldehyde 3-phosphate. Initially, the molecule is divided into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, but this molecule can be converted into GAP, and therefore you end up with two molecules of glyceraldehyde 3-phosphate. All of these reactions are reversible, and therefore gluconeogenesis can go in the reverse direction to synthesize glucose from pyruvate. The final stage of glycolysis is called the energy stage. It starts with glyceraldehyde 3-phosphate. The first reaction generates a molecule of NADH. The second reaction generates an ATP, and then the conversion of phosphoenolpyruvate to pyruvate, which is an irreversible reaction that we'll talk about more in a minute. This also generates a molecule of ATP. So this stage of glycolysis generates two molecules of ATP per molecule of glyceraldehyde 3-phosphate. However, recall that there are two molecules of glyceraldehyde 3-phosphate from each glucose. This means that each molecule of glucose that goes all the way through glycolysis to pyruvate will generate 4 ATPs. This is a 2 ATP net because recall that you spent 2 ATPs in the priming stage of glycolysis. This means that glycolysis in and of itself can generate 2 ATPs. This is with no oxygen. Of course, if you send the pyruvate into the TCA cycle, you can generate many more ATPs from the TCA cycle and oxidative phosphorylation in the mitochondria. Let's talk about the final reaction in the energy stage. That is the conversion of phosphoenolpyruvate to pyruvate. This step is catalyzed by the enzyme pyruvate kinase, and it generates an ATP. As you can see, phosphoenolpyruvate is basically a pyruvate molecule with a phosphate group added. This is an unusual reaction in that the enzyme has the word kinase in its name, but it does not add a phosphate group to the end product. It removes a phosphate group from phosphoenolpyruvate and donates that phosphate group to generate a molecule of ATP. This is an irreversible step. It's inhibited by high levels of ATP in the cell and also by alanine. We'll talk about why it's inhibited by alanine in a minute. This step is also activated by fructose 1,6-bisphosphate. This is called feed-forward activation because fructose 1,6-bisphosphate is several steps higher in the glycolysis sequence. If we go back to our overview of glycolysis, this is where fructose 1,6-bisphosphate occurs, and it can feed forward and activate this final step to keep molecules moving in this direction through glycolysis. The hormones glucagon and epinephrine can affect this step. They can phosphorylate pyruvate kinase. This leads to inactivation of pyruvate kinase, and this slows glycolysis and favors gluconeogenesis. The reason that alanine is an inhibitor of pyruvate kinase and therefore an inhibitor of the process of glycolysis is because alanine levels represent a relative lack of glucose throughout the body. When skeletal muscles are wanting for glucose, they can degrade protein to use it for energy. A byproduct of degrading protein in skeletal muscles is the amino acid alanine. The skeletal muscles will release alanine into the blood and it will go to the liver. When the liver sees high levels of alanine, it will inhibit pyruvate kinase and therefore slow down glycolysis so that the liver does not use glucose. In turn, this means the liver will then convert the alanine back into glucose via gluconeogenesis, and it can send that glucose to the muscle cells that need it. This is called the alanine cycle. So as I've shown here, muscle cells will break down protein into amino acids and they will generate alanine. The enzyme that does this, by the way, is called alanine transaminase, and you may know it as ALT. It is an enzyme that is found in the liver and is elevated when there is liver damage. The alanine then travels back to the liver where it's converted to pyruvate and then converted into glucose via gluconeogenesis, and then the glucose can go to the muscle cells which need it. But for this reason, you can understand why alanine inhibits pyruvate kinase and therefore slows down glycolysis. Once glycolysis has generated a molecule of pyruvate, that molecule can be converted to acetyl-CoA and enter the TCA cycle. And this is the way to get the maximum yield of ATP per molecule of glucose. However, if the TCA cycle is unavailable, for example, if there is a low level of oxygen present in the cell, then the pyruvate can be converted to lactate. And by doing so, the cell will regenerate the NAD that it used in glycolysis. I'll talk more about that in a minute. But what I do want to mention is that the enzyme that converts pyruvate to lactate is called lactate dehydrogenase, or LDH. And plasma elevations of this enzyme are common in a number of clinical scenarios. In hemolysis, you can detect high levels of LDH as it spills out of red blood cells that are dying. In myocardial infarction, you can find high levels of LDH in the blood, and some tumors generate high levels of LDH in the plasma. In addition, when we're trying to determine the etiology of a pleural effusion, we will often measure the level of LDH in the effusion to determine whether it's a transudate versus exudate. And I talk about this in more detail in the pulmonary modules, but for our purposes here, I just want you to recognize that this enzyme LDH has a lot of clinical relevance. Another important feature of this conversion of pyruvate to lactate is that it regenerates NAD+. Recall that we used the molecule of NAD+ in the first step of the priming stage of glycolysis. We converted it into NADH. It turns out that cells have a very limited supply of NAD+, so they need to regenerate this in order to continue conducting glycolysis, which is vital to the cell. Now, if oxygen is present, this is no problem. The NADH can go to the mitochondria and engage in oxidative phosphorylation. That will generate lots of ATP and it will regenerate the NAD. However, if O2 is not present, then the TCA cycle and oxidative phosphorylation are not available. In this case, the conversion of pyruvate to lactate is very important for regenerating the NAD molecules that the cell needs in order to keep conducting glycolysis. A very important and high-yield clinical correlation of this fact is lactic acidosis. When there's decreased oxygen around, there's less pyruvate entry into the TCA cycle. The TCA cycle becomes unavailable, but the cell needs to regenerate NAD in order to continue conducting glycolysis. Therefore, it will divert pyruvate into lactate in order to get the NAD back. This means that there will be more lactic acid production and a lactic acidosis will occur. The pH will fall, the bicarb level will fall, there'll be an elevated anion gap acidosis. And you commonly see lactic acidosis in many clinical situations. You can see it in sepsis, in bowel ischemia. Patients who have seizures can contract their muscles so vigorously that they deplete the oxygen supply in the muscles and therefore they begin producing lactic acid in order to regenerate that NAD. And this is all related to what I showed you on the previous slide, the need of the cell to regenerate NAD for glycolysis. Another clinical correlation of the need to regenerate NAD is muscle cramps. When you exercise too much, you consume too much NAD and you exceed the capacity of the TCA cycle and electron transport. You have an elevated ratio of NADH to NAD. This favors the conversion of pyruvate into lactate to get that NAD back that muscles need. This means the pH will fall in muscles and this is what leads to muscle cramps. And long distance runners have lots of mitochondria and larger mitochondria so that this doesn't happen and that they can continue to use the electron transport cycle and keep the level of lactic acid inside of their muscles low. Very rarely, children are born with a pyruvate kinase deficiency. This is an autosomal recessive disorder. The cells that are most affected are the red blood cells. They lack mitochondria, so they require glycolysis and pyruvate kinase for anaerobic metabolism. When they lose the function of pyruvate kinase, they lose glycolysis, and therefore they lose ATP. The result is that they can't maintain the integrity of their membranes, and they become deformed, and they get phagocytosed in the spleen because they have abnormal shapes. This usually presents as a newborn with extravascular hemolysis and splenomegaly. The disease severity ranges based on the enzyme activity, so babies that are completely dysfunctional in pyruvate kinase will often not survive and be stillborn. Other children are born and are generally well but can have a mild hemolytic anemia chronically. In the pulmonary module on hemoglobin, I talk about a very important regulator of oxygen uptake by red cells called 2,3-BPG, or 2,3-bisphosphoglycerate. And you should know that 2,3-bisphosphoglycerate is synthesized as an offshoot of glycolysis. When glycolysis in red blood cells reaches the stage of 1,3-bisphosphoglycerate, it can divert this molecule into 2,3-BPG using an enzyme called BPG mutase. Now, this is a big deal for red cells because they don't have mitochondria and they don't have the TCA TCA cycle, so they depend on glycolysis for ATP. And by synthesizing molecules of 2,3-BPG, they are going to sacrifice ATP from glycolysis. Essentially, they are diverting molecules away from glycolysis. This is worth it, however, for the red cell to do because some 2,3-BPG alters hemoglobin binding and helps the red cell deliver oxygen to tissues in certain settings. In the pulmonary module, I talk about how high levels of 2,3-BPG are synthesized in the setting of high altitude, and this makes it easier for red cells to offload oxygen to tissues when there is relative hypoxia, for example, when you're living in a high altitude setting of the mountains. Let's talk about how much energy a cell can derive from a molecule of glucose using glycolysis. The number of ATP that are generated depends on whether the cell has mitochondria and oxygen. The highest yield comes from cells that have both oxygen available and mitochondria. This allows pyruvate to enter the TCA cycle, and it converts pyruvate and NADH into more molecules of ATP. If a cell has both oxygen and mitochondria present, then glucose plus 6 oxygen molecules can be converted into either 30 or 32 ATP molecules in addition to carbon dioxide and water. Cells can generate 30 ATP molecules if they use the malate aspartate shuttle to get molecules into the mitochondria. They can get 32 ATP molecules if they use the glycerol 3-phosphate shuttle. And I talk about both of these in the module on oxidative phosphorylation. If there is either no oxygen present for oxidative phosphorylation, or if you're talking about a red cell which lacks mitochondria, then glucose can be converted into a net of 2 ATPs plus lactate plus water. And as I said before, red blood cells don't have mitochondria, so this process is very important for red cells. I'll finish with a couple of summary slides. Glycolysis is regulated in 3 places, and these 3 places are the irreversible steps. Hexokinase and glucokinase work right here, and that's an important regulatory step for glycolysis. Phosphofructokinase 1 works right here. It is activated by AMP and fructose 2,6-bisphosphate, as I described before. And the third irreversible step is the conversion to pyruvate by pyruvate kinase. This is inhibited by ATP and alanine, as I discussed before. And then I've also listed the 3 irreversible steps here, which are the most important steps for you to remember for step 1 of your boards. Finally, I'll finish with a summary slide showing you where ATP is expended and generated. There are 2 steps in the priming stage where ATP is utilized, and then there are 2 steps in the energy stage where ATP is generated. And that concludes our module on glycolysis.